away, Jen.
Hi, thank you. It's a great joy to be here today, and I hope I don't bore you too much. As already mentioned, I try to figure out how or whether peptides might have kickstarted the origin of life and how they were involved. And I'm giving a little bit of a some of the research that the Enigma project is doing, and this particular one was named Nickelback, not for the band, though that is a nice coincidence, of course. So if I can move on, so you all know life, like when life emerged, the early Earth looked quite different than what we see now. So we had quite a different composition of the atmosphere, and ever conditions that those were, the first organisms must have actually been dealing with this. So the energy and carbon sources that they had available were different, as well as the elements that they had available for catalysis. So when we think about the first metabolisms, we can try go back in time and look which ones are the most ancient and most likely metabolisms to have been present. And considering there was a large amount of hydrogen in the atmosphere back in the day, the one class of enzymes that is most likely to be present is hydrogenase. But at the very early stages, before life was there, all catalysis was primarily done by minerals, if at all. And right now, all we can see really from day to day are these huge machineries that are doing all this work that keeps life functioning. These large molecules are proteins made up of amino acids that are then assembling to peptide chains and then fold into very complex proteins. Now, if we think that life emerged at the beginning, these complicated machineries must have been a lot simpler than they are now. So we think that they were actually small peptides that were holding metal clusters that are still present in the final complicated protein, but they were coordinated by much smaller units that were either present before there was faithful copying mechanisms in life, or around the same time. So I'm going to talk today about about a small peptide that is emulating an activity that is in today's organism done by hydrogenases. So hydrogenases are these huge machineries with several hundred amino acids chained together. They have multiple metal cofactors. They basically catalyze the reversible oxidation of molecular hydrogen. So they can harness energy from hydrogen gas. So now they also run the other way around and have that then coupled to several other biochemical processes. And these this enzyme, this metabolic pathway, is actually very, very central today for anaerobic organisms still. Back on early Earth, we expect the oxygen not to have been present in the concentrations that we see now. So anaerobic metabolism makes sense from then. And when I started the process, when I started joining the lab, we already had like quite a lot of work done. Thanks to my colleagues, Doug, Sarosh, and Josh. They figured out on the computer how to simplify a metal cluster that does the reaction of the nickel ion hydrogenase. So it binds a nickel here in blue and an iron in orange, and how to, on the computer at least, form a small peptide cluster with similar geometries. So there was a lot of in silico work done, and we ended up with they ended up with a short 13 amino acid peptide. So now they had several candidates, and so we had to test whether those are actually working. And it turned out one in particular was actually very, very interesting. We then went ahead and tested the metal binding properties of this peptide and the coordination geometry, as well as the activity. And what we found is that throughout a wide concentration range of peptide, we ended up with 2 different species of metal-bound, nickel-bound peptide. The first species, if we give it a little bit of nickel, ends up in having 1 nickel by 2 peptides bound. And if we are adding further nickel, it refolds itself and maxes out at 2 nickel per peptide bound. So we did this over quite a wide variety of concentrations. So we believe that this most likely 2 nickel bound, as we would have expected from our computational models, actually. If we then look whether it is capable of electron transfer, what we can see is that actually electron transfer is correlated with the emergence of the two nickel species. So if you can see the more of the final species we have, the more activity we can observe in circular voltammetry, and which was quite neat, and we were quite happy about it. So the current onset potential is around 850 millivolt, which is quite negative, but it made us believe we can possibly still, if we manage to get the electrons into the peptide complex, we should be able to generate hydrogen from that. When we look a little closer into the structure, we initially wanted to determine the structure by NMR, which would have been appropriate. But what we observed is that actually the nickel that was bound in the ground state is actually paramagnetic. So we resolved to EPR for our studies as the main tool. And nickel 2+ is actually quite silent in EPR. So we reduced it to nickel 1+ in our complex. And what we found is actually a signal very, very reminiscent of the acetyl-CoA synthase. A very characteristic signal, and is a very, very ancient enzyme in itself. And that was somewhat surprising, because initially we didn't think of acetyl-CoA synthase when we tried to emulate a hydrogenase, actually. But the geometry of the nickel, of the proximal nickel, as we call it, site seems to be very similar to the one we actually designed in our hydrogenized peptide. Now, we couldn't see the second nickel that we expected to be in there. Neither could the acetyl-CoA synthase data set, actually. So that was not surprising, though not what we really wanted. So we tried to get the second nickel addressed too. And for that, we actually oxidized the nickel to nickel 3+. So when we oxidize it to nickel 3+, we see a quite different geometry. And what we can see, if we are adding imidazole as a third ligand, or a fifth ligand to the nickel, we actually see a very characteristic splitting that turns it into a very, very similar EPR signal as we can observe for nickel superoxide dismutase. So again, a very essential and very ancient enzyme as well. So what we can see is that we progressed to that, trying to do, especially for this first one, to utilize pulsed EPR to get to the ligands identity. And what we could see is that we have no nitrogen ligands close by. Unfortunately, when we try to see the nitrogen ligands for our distal nickel, we had a degrading signal. So in the moment we oxidize the system, it does some funky chemistry in here. So the similarities between the two enzymes and the hydrogenase was actually quite surprising to us. So now we did do an extensive amount of extra computational studies followed up from there. And especially Jan Siess, who's going to give a talk just after this one, did quite a bit of work and found that if you are computationally looking at the structure, the coordination that we initially thought was slightly different, but it's very consistent with the EPR data. The main problems with the EPR data and the models was actually pertaining to exchangeable and non-exchangeable hydrogens bound in the structure, but the coordination shown here in our final model is actually consistent with our data here, which is great. So we are pretty sure this is how it looks like. Now, if it comes to activity, we can also confirm we actually can convince it to make hydrogen. For this, we used an photochemical system that has been developed by Fraser Armstrong's lab years ago. And it couples basically a photoexcitable dye to our peptide via arginine, which is like it associates spontaneously with arginine, and is donating an electron upon excitation with green light. Then the electron gets replenished using a sacrificial donor, in this case TEOA. And what we can see, it's actually quite a high turnover number for such a small peptide, which made us really, really happy. The system is somewhat limited to a very short, very narrow pH range, unfortunately. So if we want to figure out whether it's actually doing this on early Earth, it's different. So first of all, we can summarize that if we bind two nickel ions, we can transfer electrons, and we can make quite a bit of hydrogen. So our active site, looking like two other essential enzymes in metabolism, is quite exciting. While we haven't quite looked at it in terms of activity, whether our peptide can in fact catalyze reactions of ACS or SOD, but that is to be done. Now, the question really arising for us now is, would this peptide complex actually be suitable to work on early Earth? And again, if you look at the conditions, our knowledge is actually limited. So in terms of temperatures, we expect it to be warmer, I would say, although direct evidence is difficult. Same with pH. We know that the carbonates and CO2 concentrations were higher. So now the question is probably not only whether it would survive early Earth, but what are the actual limits of peptides, of this peptide catalyst. So if we look at it, it is the coordination itself is pretty stable over a wide pH range. We can go from pH 5-ish over to pH 10, and it seems to not affect the folding very much, or the nickel coordination. The temperatures, we are usually working at 50 degrees Celsius to start off with, because the folding process takes some time. But we can boil it and it still looks the same. We can even leave the tube exposed to oxygen for a little bit, and it still is quite robust, which is encouraging, although not necessarily relevant for early Earth. And in terms of the onset potentials, we can also analyze it at different pHs, and we are getting basically the same characteristics on the electrochemical side. The presence of carbonate and bicarbonate also seems to be not affecting the structure very much, or the electrochemistry. However, that was actually quite an interesting and frustrating aspect, because adding bicarbonate or CN to our peptide was actually quite essential to even obtain a reduced structure, indicating that reducing the complex to nickel nickel 1 state produced very fast hydrogen, and it popped right back into nickel 2 plus and was silent. And the only way for us to trap it in a nickel 1 state was by adding bicarbonate. So it is not changing the activity as such, but made it able to, like, just about to trap it long enough for observation. Now, when we look at the EPR structures with labeled carbonate just as much as unlabeled, we cannot actually see the binding event itself. So this is supporting the presence of a second nickel, and that the ligands we are observing is the binding is on the other nickel that we are not observing, if you know what I mean. Very frustrating. So again, we have no like direct structure of a bound system, but we know it's not affecting the structure in any visible way as such. Now we tested an awful lot of conditions. This is just here a tiny subset, and we are still actually analyzing it to push the cysteine-mediated nickel binding to its utmost limits, to determine what is the cutoff where peptide metal catalysts are failing. And so far we have not determined much failing beyond actually the pH. The pH above pH 10 and below pH 5 seems to be pretty much the only way of breaking the system. So interestingly, which we haven't finished analyzing yet, is that the salinity seems to rescue the nickel binding event at low pHs to a certain extent. So I know it's like really difficult to discern in this raw data, but it's basically if you have really ultra high concentrations of salt, the binding of nickel is a lot more stable, and you can push the pH a lot lower. But yes, this is like fresh data, and the electrochemistry is not analyzed at all yet. So I have to apologize for this. But just to complete the challenge of our little peptide, we actually threw it into Jersey Shore water. Whatever is floating in there seems not to actually shock the peptide at all. It's still perfectly happy coordinating nickel. As long as you keep the pH stable, it's behaving just as normal as if it is in a clean buffer. So all the random organics floating around, which now means it is we're pretty sure that on early Earth, as long as the pH was in that suitable range, our peptide complex could very well have existed and functioned the way it does in our test tubes. And we are also sure it's forming a homogeneous species, like particle species, and not some mixture or aggregation. So it's a very, very robust system, which is very, very encouraging considering early Earth. We are limited in our knowledge about early Earth. Now, further questions, of course, they're just about 1 million and more. And a lot of them are still questioning what is the limitation? What is the limit of how far we can stretch metal peptide catalysts. Do different metals, like we have done all our studies with nickel so far, like can other metals be just as robust as our nickel binding, or will there be differences depending on which metal or which amino acids do the coordinations of metals? Will substrate binding be the main weakness of the peptide catalysts, or what are the limitations? Now, what we cannot really address in our current setup is the pressure to test higher pressures that might be present on different environments on early Earth, as well as on other planets. That is a little more tricky and needs further addressing, whether there are problems with the system. And then the questions specifically about the hydrogenase activity that we are seeing is pertains to the mechanism. Where do those substrate protons come from? Where are they bound? And preliminary data seems to indicate here that it seems to be cysteine protonation that is determining the activity or productivity, but it's something that requires certainly further investigation. And as I said, like there are a million other questions that I haven't even thought about yet. And we are very, very glad that we have such a good team working on this project, really. So I have to thank a bunch of people that have worked on the project and contributed and continue to contribute, and great collaborators as well. And I thank you. And if you have any questions, please be free to hit me.
All right, thank you so much for that really interesting talk. So it looks like our second speaker, Jan, hasn't been able to make it, so I'm going to revise our plan a little bit. And if Jen, you're okay with it, we'll jump into questions for you now and see if he can make it later. Else we'll just have a short session today. Oh, okay, so we have someone, Bay. Beep?
That is not B, but yes.
Okay.
George. Hi, George.
Okay. All right, so I see the chat's going. I could sort of.
Yes. So yes, it's a good question. So yes, we are more about life on early Earth right now, but the application to clean fuels is quite relevant, of course. So on early Earth, in order to harness energy from atmospheric hydrogen, we would run the enzyme technically in the other direction. But for assay reasons, and in our case, we run it the way that we can harness that hydrogen as fuel, or potentially. We have done a couple of experiments like to optimize. It's not quite the amount of hydrogen we would want to have for industrial purposes, so it's not that great yet, but there is certainly room for improvement. So with mutation and further improvement, I'm sure you could optimize that. And yes, I have tried to crystallize it for diffraction, and it has so far refused. We have fused it to crystallization tags, and it's very good at preventing crystallization. So I'm, as a protein crystallographer, I'm very frustrated by this, incredibly frustrated. But it's ongoing as well. So we have now like a different crystallization tag that hopefully works for us. But yeah, it's ongoing.
And just to recap for the online recording, when people won't see the chat, the two questions were whether this could be used for fuels potentially, and whether the peptide could be crystallized. And then we have a question from Alex about whether you have speculated about potential sources for the donor electrons on early Earth?
Yes, we have. One obvious source of like general energy is the sun, so UV radiation. It will have to be coupled with something else in order to harness that energy, most likely. And we had a couple of ideas about a potential combination with mineral surfaces, but we haven't gotten very far into the investigation quite yet. But that's another, organic molecules as well as mineral surfaces may very well have donated the electrons.
All right, it looks like our second speaker is online. We didn't recognize the name, so we'll get him on board. But for now, I'll just, while it's here in the chat, I'll ask you one more question about the metal binding. So with the peptide motif you have, do you expect nickel to outcompete other metals?
So I'm doing those experiments actually as we speak. It's quite funny. It really hates iron. I tried to push iron into it and it's just not having it. It will bind cobalt. I have not finished that experiment yet in terms of like how they are competing against each other. I mean, both of them bind independently from each other, but we have not observed any activity as such. So I'm not a geochemist, so I can't really hypothesize about the abundance of cobalt in the early ocean, to be honest. I'd be curious. I need to look that up actually. But it's very sure it likes nickel. In terms of zinc, I'm a little terrified, though the binding environment of zinc is primarily tetrahedral. And in our computational studies, tetrahedral coordination is quite strained and it doesn't really appreciate that very much. Though if you imagine a scenario where you have like a dimerization happening, then that is a possibility. But we have so far focused entirely on nickel and a little bit of cobalt. It does bind copper too, but also not as happily as it is currently with nickel.
Becca, the second speaker is here.
Yes.
He's under one of the Bay beerers.
Yes, so we'll see if we can get him online. This was a great bonus question session. But let's see.
I mean, if he wants to use my ID, like I can send him a link. Then he can be a second Jennifer Timm.
I think we have him marked as a co-host now. So Jan, could you try sharing your screen, unmuting, putting on your video? We'll see if any of that works. I think we can hear you and we can see you.
Yes, I'm sorry to disappoint. I'm not B. That is not I. No.
We're not disappointed. Right, if you can share your screen, I'll go ahead and give your introduction.
Perfect. Yeah. To the real B out there, I'm sorry. I have no idea what happened.
All right. So we have with us Jan, who is a graduate research assistant working in the lab of Dr. Vikas Nanda at Rutgers University. He currently employs various computational procedures to better understand the mechanisms governing evolution and the origins of life. Jan's research aims to improve the mapping of phylogenetic trees that extended to the ancient past by using molecular dynamics of proteins as an additional metric. His work has merited significant recognition, including the Rutgers Presidential Fellowship and a Fulbright Award. So we can't see your screen. If you can make it full screen, then we're good to go.
Okay, can you see it now?
It's processing for me. I think it's almost there.
Okay.
You put it into full screen mode?
Yeah, now it's in the full screen mode.
I'm still seeing the more PowerPoint version.
Oh,
It's plenty big enough for me.
who can start sharing with some of the?
I think maybe you just need to switch views, maybe?
Is like this?
Uh, kinda. Now we see your version. There you go.
That's perfect.
Okay,
Alright.
beautiful.
Go for it. Thank you.
Yeah, of course. So after that little bit of juggling right there, thanks. I'm Jan, not B. It sounds similar to when you're really tired. And I'm going to be getting into a rather truncated kind of version of the work I've been doing in utilizing dynamical attributes to parse Ferrodoxin structural domains. So naturally, just jumping right into it, I think the first thing that you'd ask is, what is a Ferrodoxin? And for those who don't know, they're these small oxidant reductases, and they bind iron sulfur clusters of varying stoichiometry. So 2 iron, 2 sulfur, 4 iron, 4 sulfur clusters. And they're ubiquitous across all of the domains of life. So they, the bacteria, eukarya, archaea, and they perform some of the most important functions in maintaining life on all of Earth. So electron transfer, and they are involved in facilitating that process. They play important roles in redox reaction, the metabolism, photosynthesis, respiration, nitrogen fixation. And they're also highly conserved. So they potentially could make a very good phylogenetic marker too in evolutionary studies as well. When you're looking at their sequences, so I focus primarily on bacterial ferrodoxins, the domain classically looks like, are often they're quite small. They range from about 55 to 60 amino acids, sometimes 100. And depending on the cluster it binds, it could have a very specific canonical binding motif. Which in this case, for 4 iron 4 sulfur binding ferrodoxins is the CXXC, XXC, and then some number of X's after that, CP. And I've underlined the motifs in red. I'm sorry, the C and the CPs are where the cysteines and the prolines are. In the pink and the cyan is what I want to highlight here, is in which in this paper published by Margaret Dayhoff, a famous biochemist, and Dr. Eck, they took 2 halves of this ferrodoxin, the 2 halves of this ferrodoxin sequence, they split it in half. And what you see is that there's actually a high degree of overlap between them. And so what this brings into question is, well, if I'm going through this one by one and I have an overlap in row 4, which is the result or the similarities between these two, something interesting is going on, because I can't really chalk this up to chance. I mean, you have 2 to 3 similarities that would come up, that would be chance. But in this case, we're having about roughly 12 matches. So something else is going on here, which is pretty interesting. And I'm going to get into a little bit more later, what was derived by Dr. Dayhoff and others. Additionally, you also see ferrodoxins being incorporated into these larger macromolecular machines. So what I have here is this a PDB-ID 5T5I, and I've made it a point to highlight these iron sulfur clusters, which form this pseudo wire of sorts. And in that, onto the right-hand side, I've also isolated what it looks like in its cartoon representation. So my particular fascination here is not really just, I mean, this whole structure, of course, it's interesting, but I'm interested in more of like what these ancient parts or modules really looked like. Ferrodoxin being one of these predominant large parts in this parts list. So my lab's, one of the big hypotheses in my lab is that the earliest known proteins were those that were involved in electron transfer. And of course, the reason for that is because every living system that we know of on Earth is involved and partakes in this process. So if you have these large oxidative reductases, the idea is that they're too complex to have originated as is. Ideally, or what makes more sense actually is that they would have come together and have originated from these simpler parts. And while I, in this presentation, focus mostly on already the parts that have been isolated, I want to make a highlight. I want to mention that my lab has already done a lot of work in this previously, where they've clustered and created a large list of these fundamental parts in these larger machines, where you take about 9,500 structures, for example, you align them and then create these microenvironments, which consist of 15 angstrom spherical radius from the metal, or one of the metal centers or multiple metal centers as they appear in the structures. And then classify them in these microenvironments, and then from there, with some other magic, so to speak, come up with these modules. One of these predominant or major modules that show up, of course, are these ferrodoxins, the symmetrins, and so on. So I just wanted to mention this briefly, but I focus primarily on these smaller ferrodoxin parts. So there's some evidence to suggest that, not some, there's a good amount of evidence to suggest that each half has undergone some independent evolution. So I mentioned Dayhoff's work in the previous two slides, and I make a point to mention this the sequence again in point A, where you see these two halves instead now just colored in blue and gray. And they belong to Clostridium pastoranium, which was originally analyzed. And the iron sulfur clusters are in the orange and the yellow. So what you have is an overlay of these two halves to highlight the high degree of the internal symmetry that's going on between the two. It's almost like they're identical when you overlay them on top of one another. And when you go below that, you'll see that there's this phylogenetic tree. And the idea here is that you have lineages that are colored according to the internal symmetry and the structure of the inferred ancestral nodes that preceded. And this analysis indicates that although it's derived essentially from one sequence, you have both suppositions that were proposed by Dayhoff, which ended up being correct. And the first is that the identity is the sequence identity is increasing, and the more that you go back in time. So from this, I don't know if you can see my mouse, but when you go from the right-hand side to the left-hand side, it shows about a 59% internal identity compared to an average of about 27% in the contemporary sequences located on the right. And the second is that the fraction of these abiotically synthesized amino acids is also higher in the inferred ancestor, which you can also see being this inferred red ancestor on the bottom of this. I'm having a problem again there, but anyway, on the right-hand side, you'll see that there's also another a maximum likelihood phylogenetic tree showing effectively three clades, where the first clade in gray is consisting of both the N and C terminal halves of the ferrodoxin, and from the blue, the C terminal, and then finally the last one would be the N terminal one. And when you compare their similarities with this tree, it's clear to see that independent evolution is taking place for each of these individual folds, as evidenced by the separation of these clades. So in all, this really supports this notion of the independent evolution of these variable positions within the ferrodoxin, and the idea that an ancestral duplication event had to occur. And kind of going off of that idea, in addition, is this idea of recent works that I've been doing the last year with a colleague of mine named Banu, in which you have this ferrodoxin that had been split from the connecting loop region, as evidenced by this dashed line. So you have 30 ferrodoxin, 30 amino acid long halves. And what we find is that they assemble not only as dimers, but they also bind these iron sulfur clusters. When you go to B, you'll see that there's an FPLC chromatogram of these species, and they separate out very similar, and indeed are showing this pattern that they are dimers. When you look below and see this UV-Vis absorption spectrum, you'll have these characteristic peaks around 370 to 380, which is characteristic of iron sulfur binding. And indeed, you could use EPR to look at paramagnetic species to display signatures that are characteristic again of this binding. So their spectra are very similar, and we could argue that the domain in this instance isn't really a 60 amino, classically referred to as like a one whole unit, or this 60 amino acid ferrodoxin, but it could potentially be these two individual 30 amino acid halves instead. So I talk about I talk about using dynamics, and some people might ask me then, well, you've used all of these sequence-based methods in the, already. Your lab has done this. Why don't you just continue along with this? And in a sense, I'm not completely neglecting sequence. Sequence is a pivotal part of my workflow, but I think it's important in trying to find out what this true or what the real domain and Ferrodoxin, I mean, of what the real domain is, or what these domains are in these larger sequences area you want to study, the most ancient part of it, that using these statistical models, which is essentially what these with phylogenetics and these other trees, tree construction algorithms are, the further back in time you go, the more susceptible you are to having bad data, essentially, or untrustworthy results. And this is due to the a slew of mutations that it can occur and accumulate in these sequences with the substitutions, insertions, and deletions. And they can be so extensive that it could completely change the original sequence, and it could be no longer reliably inferred, essentially. So when you look at the context of deep time, millions, billions of years, in this case, 100 million years is all it takes for results to be to be bad or tainted in a sense. You begin to encounter a lot of limitations and challenges. The other thing is that when you start to use solely sequence-based methods, while you do have the potential to accumulate a large density of data, when you put it through your algorithm, and you might need to filter it based off of some arbitrary threshold, you might actually get rid of a lot of sequences that might be homologous in the first place. So what I have here is concavalin and peanut lectin. And the idea here is that topologically speaking, they look very similar, and have undergone what's called a circular permutation. So if you were to feed this through some sequence algorithm, it might not actually say that these two are homologous or similar at all, due to the fact that there's some rearrangement in their sequence due to maybe fission or fusion or some other duplication event. But in fact, they appear to be at least analogous to one another. And if you were to use structure, then you start to also encounter some more problems, because you can have something on the left-hand side that looks like a ferrodoxin, but is actually just something that spans something that was extracted from a larger protein that could be about 1,000 amino acids long, and it's just an insertion. So you could have completely two far away pieces that are coming together and just look like they're a ferrodoxin. And then conversely, on the right-hand side, you have something that is also another insertion, but it's inserted into another part of a protein, looks like a ferrodoxin, but it's missing its second iron sulfur cluster. So I guess the real question is, why dynamics then? And one of the answers to that is because dynamics has been proven to be shared within a family of proteins. Of course, this isn't ferrodoxins, but it extends to globins. And you could apply a method that decomposes, I guess you could say, in a structure into a series of masses and springs. And you could model dynamics based on the interplay between these two nodes, or these alpha carbons, for example, and how they move in relation to one another, or deform, or react in the face of some deformation. And you can plot out their you call them modes, or the way that these movements manifest as a result of this analysis. And when you overlap their fluctuations, you can actually see that there's a high degree of similarity between the first 4, 5, 6 modes. And when you actually do this with ferrodoxins, in this case, 3, 4, about 8 homologous ferrodoxin, you can see that this also maintains the same sort of idea here as well. They're colored differently because I just clustered them based on the similarity of each of these fluctuation profiles. But you can still see that there's this characteristic large fluctuation around residues 28 to about 30 or so. And this is the loop region that connects those two ferrodoxin halves together. But otherwise, you can see that they still follow a very similar pattern in terms of the way that they're amino acids fluctuate. And more recently, actually, just as of yesterday, I was able to figure out a way to, motivated by a colleague's work where he tiles structures in a windowed fashion. You could take a sliding window of a cross-correlation matrix, in this case, which refers to the way in which you have one amino acid moving in a correlated or anti-correlated fashion amongst all other amino acids within a similar portion of sequence, and effectively see if they're using a metric, a similarity metric called this cosine similarity. You could see if the movement of the portions or indices, portions of sequence corresponding to these lengths and these tiles are moving in the same direction or not. So if you were to look, my mouse is not working, but at around like 11, or a tile center index in this X portion here, if you were to be centered around 11, you have a tile length of about 6, for example, that spans 6 residues long, but it'll, according to the numbering conventions used in your PDB, go through the middle, or have its middle portion be at 10, residue 10, for example. And considering this is using alpha carbons, it would be at the 10th, that 10th alpha carbon there, or going through it. So 3, 3. And we could see here, this is the 2-FDN, very similar to the one, the ferrodoxin that was used by Dr. Dayhoff, that you have this asymmetric pattern on the left-hand side and the right-hand side, showing you that there are these very specific portions of sequence that, when tiled or tessellated across the other portions of sequence within itself, it exhibits, some portions at least, a high degree of similarity in relation to other portions of sequence. So around 10 would be this, around 10 would be probably, oh, there it is. This portion where it's binding, the loop that binds this iron sulfur cluster. Ultimately, what I'm trying to do with this is to create a dynamics fingerprint. So I have some homologous ferrodoxins here, all belonging to the same family. They're these bacterial ferrodoxins. And what I want to do is I want to create a domain fingerprint, or a dynamical fingerprint, that I could then use to apply this windowing sort of approach, this tessellation approach, to larger proteins like this 5T5I that I showed you all in the beginning, to better identify domains, ferrodoxin domains within these structures. And this could be used as a scaffold for further protein engineering endeavors or for identification of the boundaries as well. In addition to a host of other things. And so with that, I'd like to thank you, not only my lab here, but for all of you for listening and for hosting me and for allowing me the time to speak. Thank you.
Yes, and thank you for that great talk. So we're going to bounce back into questions. Questions for both our speakers, although since we gave Jen some time for questions before, we can maybe, I won't be hard on this, but focus on some questions for Jan first. So the chat should still be open. And yeah, it appears to be working. All right, so
I have a question.
go for it.
Hey, Albert Fehrenbach here. Thanks for the really great talk. So this question is for Jan, but I guess could apply to Jen too. Do you? It seems like you have a methodology maybe to try to point out which were some of the first domains to evolve or otherwise, arise through prebiotic chemistry, even at some later stage. Is that true? I mean, is there any way we can identify what the first sort of functional domains were?
So I think that's very I know with my method, I know that, for example, considering the list of ferrodoxins, you can feed it through an algorithm that takes these windows, essentially, these portions of sequence that are moving similar when you compare it to the other portions of itself, and you could create a portion of it that maximizes the score, and what that'll give you is some final sequence of a particular length, and you'll see that it creates a half ferrodoxin, but a very primitive appearing one that consists of only the cysteines binding the iron sulfur cluster and the alpha helix at the end, toward the end. As to whether or not this is going to be like an ancient ferrodoxin, I think more work on my end needs to be done, at least. And I don't know exactly how to even answer that after I even extract it.
Yeah, right. Okay. Thank you.
Thank you, though.
I also have a question, which is, you have the dynamics down for ferrodoxin today, and ferrodoxin today is functional. Is it correct that you're trying to look back? Maybe this was Albert's question in a way, but look back at different nodes in evolution? And if the answer is yes, whether you'll have any idea based on the computed dynamics, whether those would be functional?
Oh, I see. So in a way, I'm trying to use dynamics and that information to circumvent the limitations that we have with these statistical protocols and the whole the molecular clock, for example. I know for a fact that the structure evolves about 3 to 5 times slower than the sequence. It's been published. And you're selecting for function, which would hopefully manifest in this dynamics. I'm trying to find ancient ferrodoxins or more primitive ferrodoxins by use of these dynamics, effectively trying to go back further.
With your dynamics, the sequence that corresponds to some sort of dynamic pattern. Is there any idea to take that sequence and test it in the lab and see if it has activity?
Yeah, so I did. So in the original version of this talk, my lab published a section about these microenvironments where these parts, and I made mention of a parts list. And the idea, a lot of what motivated this too, is that when you look at the so the main assumption is that the oldest parts of the proteins within that parts list centered around those cofactors that have been bound. So when you actually look at the structure of this portion, this 15 within this 15 angstrom sphere where the metal constitutes the center of it, you'll find that these domains or these microenvironments environments are discontinuous in sequence. And it doesn't abide by this classical definition of a domain where you have something that is the opposite of that. So the idea is to, definitely like create the extract portion or identify these domains, and with those sequences that we have been able to come up with, then test it in the lab. But we can't really do that well if we have these discontinuous portions that have been extracted from the database. We kind of want to refine that.
Okay, that's a great answer. There's a comment in the chat from Hanadi that I'll read. It says, I think that the secondary and tertiary structure of a protein rather than its amino acid sequence is way more important, especially in origin of life scenario. Can this fluctuation dynamics approach help us to examine proteins with this perspective? Okay, so yes, there was a question in there.
I'd scroll a bit. The tertiary structure of a protein rather. I do think so. Yeah. And that's also the case. So I think there's a limitation in this way as well, because I think that there's some protein or some amino acids that would be resistant to mutation, but there's only a point or an extent where things are going to go wrong, where there's certain parts where you can't mutate them. And unless, like, if you were to get rid of the cysteines, for example, I think hell would break loose. So I think that it could help to an extent.
And there's some follow-up on that from AC saying certain amino acids are crucial for function, whereas other amino acids are more structural. If you have a comment on that comment.
Oh, I think I made it a point. Yeah, no, I think I tried to echo that. I think toward the end, actually, by saying like, oh, there's certain, like, if I were to get rid of my cysteines, well, we're not in for a good time.
Yeah. All right. So any questions for both speakers?
Thank you so much for the good questions.
I have another question.
Go for it.
This one's for Jen. First of all, very nice talk. It was amazing what you've done. I was just wondering, do you have an idea of the binding constants to the two nickels? Are
No.
you? Yeah. Oh no. Okay.
Short answer. No. So the problem. So we haven't run any ITC, which would have been the technique to address at least the first binding constant. It is in the low nanomolars because we see for the first binding event. So we have these two species. So the first binding event happens and maxes out at the ratio of 1 nickel shared between two peptide. And that one, when we do the titrations at low micromolar, we already see this, and we would expect actually to be even lower. It's like an instant binding event. So the problem we have with the second binding or folding event is that it is incredibly slow. So just to give you an idea, is like I'm doing the titration with metals right now as well, and it's you give it the metal, and it takes 50 Celsius overnight at least to get anywhere. So anywhere near completion. So it is a very, very slow process. So to get like data for the ITC, we would have to run it at high temperatures or for an incredibly long time. It's very slow equilibration. So that has been so far making our life a little bit worse, unfortunately. But yeah, the first binding event is quite high affinity. The second we can't really tell.
Oh, okay. I was thinking you might be able to just extract it from the data that you already have, like through in the CD titrations that you did.
Our maximum or minimum, I need to look that up whether I can extract that, because our binding or the minimum concentrations of the peptide that we can run and not sacrifice our signal is still like in the 15 micromolar is probably the lowest concentration we can use the peptide at, and that is still kind of like higher than we would want to, because I'm pretty sure the affinity is higher or better than that. And so CD is like somewhat of a limitation if it comes to sensitivity, unfortunately.
Yeah, right.
It also gets actually significantly slower. So if I'm actually lowering the concentration of the peptide, the ratios are the same. It's just like the transitions are getting even slower. So even at 50 degrees, we are talking about all of a sudden several days of equilibration. So it's getting like a little bit painful. But yeah, I will have a look whether the kinetics, the folding and binding kinetics can give us some more information about that. We usually record several time courses throughout, so we can try and squeeze it.
Interesting.
Thank you.
And just to follow up to that, is it completely selective for nickel, or you might have mentioned this in your talk, I missed it, but do other metals also bind?
So because the main technique we are using is CD. We don't really have a good way to observe zinc very well, because zinc is silent in CD. But it certainly binds cobalt, also in a significantly different way. So we can disentangle those signatures quite easily. It will bind copper, and it will not take iron. So the problem is not that whether it is binding. I mean, any cysteine environment is going to bind multiple different metals. The problem is really stability and geometry in the end. And to determine the geometry without running EPR experiments or like further spectroscopies is not that easy. So we know we can bind things, but they are not the same geometry. So for example, copper, we showed that, I didn't show it actually, but we have data showing that it also has a similar, it also has 2 copper per peptide, but the distances of the 2 coppers are significantly different, like a lot wider. So it's it binds, but it's not the same kind of binding.
Yes. Yeah. And one more question, if I may.
Yeah.
So the nickels are very close together in the structure. Do you see any evidence for like a mixed valence state during the reduction process for the 2 nickels? Just out of curiosity.
We should have seen something like that in EPR, and we didn't. So yeah, we don't have evidence for that. And so there is a certain, so if you have the 2 nickel and you look where, like what is bound to those nickel, and so if we use the reduced one, we can in pulse DPR, we should be seeing nitrogen ligands if they are available. So now none of our models contains nitrogen ligands as such. However, the data is telling us very, very weak association of nitrogen ligand. So one option is that it is one of the amides adjacent to the coordinating residue, or it is in fact a 5% bleed over throughout or relayed signal we getting from the second nickel that is coordinated by two amides. But the data is not strong enough for us to make this claim that there is like a 5% sharing of the electron clouds
Right.
of the two nickel. We like there's no proof, so we wouldn't necessarily like go out and say it, but we have data that would be consistent with something like that, but not beyond like something like 5% sharing.
Ah, interesting.
So,
yeah.
Otherwise, we would have seen a lot stronger signal from the other nickel.
Yeah. Very cool.
But it's a very good question.
Yeah, thank you. Thanks for the nice talk.
Thank you.
I'll jump in with a question for both speakers, actually, which will give anyone else a chance to think of questions before we close out. Darnish, do you have a question? Maybe not. All right. So my question is, well, both of your researches seem to be developing a workflow that could be applied to other small peptides or other proteins we're interested in their ancient history. So with your workflow, what would be next if you were to investigate another system or another sort of set of proteins or peptides?
The interjection. So earlier in the talk, I guess like in the middle of the talk, I cited a reference to where somebody had applied this to globins, to the globin family. So I don't think this is exclusive only to ferrodoxins, for example. If anything, I think it was interesting to see that this was replicated within ferrodoxins because it had already been proven with the globins. So I think based on that, there's application to other families as well. As to where this goes, I've been trying to figure out specifically with the heat map what that means. Like, we have pretty colors. What does rainbow mean? And to me, it seems after applying, you could say it's an implementation of dynamic programming in some way. You get a portion of sequence corresponding to a particular movement or a vibrational mode. And this could be a portion of sequence that's it could be a start of there could be the begin. I guess with this movement or with all movements summed together, it could be maybe a starting scaffold for maybe other protein engineering efforts, for example, because it seems to be shared. Maybe not only in this structure, but if it appears in other ones, it could be something you could use for other efforts. I think my bent's more on the evolution to see if maybe this is something that could be used. Is this the starting structure? What is the smallest part we can get from this?
Yeah, I would just add, yes, I mean, one obvious thing is to try whether all enzymes, all metabolically important enzymes can be reduced as far as we currently reduce our hydrogenase, right? So where's the cutoff? How small can we define a unit? And what could be the origin of peptide or of proteins? Like how could they have gotten bigger? So it's like an evolutionary trajectory, but also the interplay between the different enzymes. So obviously, like one enzyme or catalyst doesn't make a metabolism, and so there is a lot of like what is part of the direction we want to take it is to figure out what are the electron sources? Can we couple multiple, or could there be a spontaneous coupling of multiple different units that are associating with each other, and then having electron transport chains to lead to more complex and more electronically demanding reactions. So like to have like artificial electron chains emulating metabolic processes in cells. So like almost like wiring diagrams, that's where it's going as well. So on the catalysis side, an individual, but also like if evolution happened to make things bigger, and why and how and what can be done with it.
Yeah, those are all really interesting questions. All right, so it looks like the chat has settled down. So if you have any last questions, get them in now. But I'll just remind everyone that we do this every 3 weeks, same time. And we also have a Slack that if anyone wants to keep in contact that way, I can go ahead and pop the Slack link into the chat. But with that said, thank you again to both of our speakers for your excellent talks, really interesting talks. We loved hearing from you and your time, and we're getting thank yous from the chat as well. So as soon as
thank you for having us. It's great to talk with so many good people and getting good feedback or like questions. That's invaluable.
I echo the same sentiment.
Yeah, thank you. So with that, we can sign off, and thank you again, everyone.
Thank you.
Thank you again. Take